at Center of Ecological Science, ISC Bangalore. He is founder of Evolutionary Venomics Lab and director of Venomic Institute of Snakebite Health Lab and director of Advanced Medicines. His group is working towards developing anti-snakebite therapies for India and sub-Saharan Africa. Stage is now all yours, sir. Please take it.
Is mic working? Can you hear me? Yeah, great. Hi, good afternoon. I know it's the fourth talk of the day, and just before the tea session. But I think I have something that will keep you awake better than tea: snakes. So I'll talk about snakes and the venoms. And I hope this talk doesn't give you nightmares, because I'll be showing a lot of pictures of snakes. In fact, I hope that it has a opposite effect. Yeah, so just a brief background on my lab, the Evolutionary Venomics Lab at the Indian Institute of Science, Bangalore. So we are interested in all kinds of venomous animals. We are interested in three major aspects of them: understanding how ecology and evolution influences their, the venom cocktail, the potency and functions, and we are interested in, developing better therapies for treating this disease called snakebite, which all of you would have encountered or heard about, but it's one of the most largely neglected of the neglected tropical diseases. So you'll see that in a couple of slides. And we don't work on just snakes, we work on all kinds of venomous animals, scorpions, spiders, these endangered mammals from Cuba and Hispaniola, which haven't changed since the dawn of the dinosaurs. They're also venomous, there are lots of venomous mammals. So we work on all kinds of venomous animals around the world, and not just in India. After the presidential lecture, I had to include this slide and tell you that we also use CRISPR-Cas9 to manipulate the genomes of venomous animals to study how venoms are produced, and venom-producing cells evolve across time. So we looked at these animals called sea anemones, which belong to the same phylum as cnidarian, as jellyfish and corals. So they have these stinging cells, unique stinging cells, which shoot out and inject venom. But we didn't know how they originate and how they diversify, and so we made transgenic lines of these animals so that they produce fluorescent protein only in these cells. That's how we could isolate them and look at how this transition happens. But in my talk today, in the interest of time, I'll be largely focusing on venomous snakes, and I'll also be showing you a lot of pictures that I've taken of venomous snakes from India and around the world. So that's a beautiful soft-scale viper. So you have all seen snakes. They belong to largely into these four major families: boas and pythons. They are considered non-venomous, right? And then you have elapids, which includes cobras, kraits, sea snakes, king cobras, and then you have all the vipers in the world, right? So these are the two venomous groups. And then you have a third group, a fourth group, which is often called a non-venomous snake, right? Which includes snakes like vine snakes or tree snakes. Are there any snake enthusiasts in the audience? Do you guys know snakes? So what is it? Is it a venomous snake or a non-venomous snake? Thank you. Thanks for using that term, right? So if you ask herpetologists, they call this a mildly venomous snake because when it bites, you don't die, but you suffer from symptoms, right? You have swelling and you have bleeding. So that's why it's called a mildly venomous snake, right? But venom is a complex cocktail which is produced by one animal to inject into a target animal in order to facilitate its feeding or defense, right? It's using venom to either feed or defend itself. So venom is like pregnancy. You can't be mildly pregnant, right? So similarly, venomous animals are either venomous or non-venomous. There's no third grade of being a mildly venomous animal. And I'll tell you why they were called mildly venomous. When people looked at secretions of these tree snakes, they found this thing called three-finger toxin. It's called so because it looks like three stretched out fingers. And this is a toxin that makes the venoms of cobras and kraits so dangerous. You'll see that in a few slides. And so when they found them here, they assumed that these snakes should also be very toxic. But when they tested them on mice, they didn't kill mice. They didn't cause a lot of effect. That's why they called them as weak three-finger toxins. And of course, if something doesn't work on mice, it's unlikely that it will work on a human, right? But when they tested the same venom cocktail on the natural prey of these snakes, which are birds and lizards, which is what these snakes feed on, it works as efficiently as a cobra venom would work on you or any other mammal, right? So venom evolves to target, the target species. It doesn't have to have any effect on humans. So saw-scale vipers. If you go to South India, my part of the world, you will see that you have these very tiny snakes called saw-scale vipers, and they're extremely potent against mammals. So this is LD50, right? The smaller the bar, greater the potency of the venom, right? So they are extremely potent against mice. But if you go to Gujarat, not too far away from here, you'll find really big saw-scale vipers. They are almost as big as Russell's vipers, but the venoms are not very toxic against mice, and that's because these snakes feed on scorpions. Similarly, kraits. Kraits are supposed to be one of the most toxic snakes in the world. Look at the potency of the venom; it's extremely high. But if you go to eastern India, West Bengal, you'll see that the venom potency is really low against mammals, and that's because these snakes feed on other snakes. They don't feed on mammals as much. And then we have another species of snake called a syncrate, which is originally was found in the Sindh region, but we have discovered them further down south as well. Look at the potency of the venom. We don't even inject the venom into mice; we just show them and they die. Right? The venom is that toxic, and that's because in captivity, when we try to feed them, kraits are voracious snake eaters. They eat any snake that they encounter, but these guys refuse to feed on any other snake. They just fed on mice. So this shows that ecology underpins venom evolution, and humans have nothing to do with it, right? And then boas and pythons, so something that I called non-venomous. When we looked at their saliva, we actually found three-finger toxins, the same toxins that we find in cobras and kraits, right? So they are not venomous, but there are many species of boas and pythons. If you go to Australia, you'll find these black-headed pythons, and if you see how they feed, they don't strangulate their prey; they actually bite them repeatedly. So there's a lot of things that we still need to understand about the origin of venom, but I'll not get into any of this in the interest of time. And we'll move on to snake bite, which is caused by venomous snakes. And this is a major concern in this part of the world because over 58,000 people die every single year from snake bites in India alone, and that accounts for over 50% of global deaths due to snake bites, and 3 times the number suffer from permanent, immutable injuries. And this is a major problem. Unlike cancer, which doesn't discriminate between the rich and the poor, snake bites do, and they disproportionately affect people in the rural areas because these are farmers and laborers. It's an occupational hazard for them, and that's why it's nobody cares about snake bites, and very limited resources are being spent on snake bites to discover better therapies, which I'll show you why we need them. So snake bites are treated using antivenoms, which are produced by injecting venom into horses, ponies, mules, and then collecting the resultant antibodies. And we have shown in the past that venoms can vary even within the same species across geographical regions. So one of these rattlesnake populations in the US was extremely neurotoxic, but the antivenom was being made using these, so it won't work against that particular population, right? But ignoring all that, we still produce antivenoms by collecting venoms from just one point location in southern India, and this antivenom is used for treating snake bites all across India, and unfortunately even in Africa until very recently. And we know that these antivenoms don't even work in Karnataka, where my lab is. I mean, they don't work very efficiently. They do save lives, and that's the only way for treating snake bites as of now. But it has a lot of problems, including this limited, species specificity. It only works against the species and populations which are involved in, from whom you collect venoms for antivenom manufacture. And then this technology has not evolved in over 100 years, which means that if you take a vial of antivenom, almost 90% of this vial is redundant. It's therapeutically useless. Only 10% target snake venoms. And as I showed you, we don't have to neutralize all snake venom components. There are only a few that affects humans. So there is limited dose efficacy, and that increases cost of treatment. In some places, antivenom vials, a single vial can cost close to $100, which these people cannot afford. And finally, because these are horse antibodies, and if you inject large amounts of these into humans, you can imagine that it can cause secondary reactions. Sometimes fate can be fatal, like anaphylaxis. You see that very often. So they are not safe in the current form. This is why we need better strategies. And before I tell you how we are trying to introduce those better strategies, just a background on snake bites. So there are four major snake species that are responsible for causing snake bite deaths in India. These are cobras, Russell swipers, kraits, and then saw-scale vipers. And what we have been doing is we have been going around the country to to all kinds of locations and collecting venoms of these snakes, and then we bring them back to the lab and then we analyze. We collect them from dense forests to deserted places, and we collect venoms from not just the big four snakes but a lot of other medically important snakes against which no antivenom exists today. Right? So we don't focus on just the big four but also on other kinds of snakes. And I'll show you now how much variation you can find in snake venoms and why this has become a major challenge for everyone. Despite all the advanced technologies, why is it a very difficult problem to solve? So there is a lot of variation, as I had showed you, in snakes abroad. So let's look at cobras from different parts of India. This is a study that we published a few years ago, so this is not updated, but we have venoms of these snakes from all corners of India. And so this is like a fingerprint of the venom, right? So if you focus on peak number 4, which is what contains the dreaded three-finger toxin that I told you about, look at some populations like Punjab and West Bengal. They have really large amounts of that toxin, right? But if you go to the neighboring state of Punjab, Rajasthan, you'll see very limited amount of that toxin exists there. And if you mass spec and see what is there in the venom, you'll see that almost 80% of this Punjab venom is made up of this neurotoxic three-finger toxin, causes neuromuscular paralysis. People can't breathe. That's how they die, right? And if you go to if you go to Rajasthan, only a small amount of that is neurotoxic. Large amount is cytotoxic or cardiotoxic. So a lot of people who suffer from cobra bites in these regions, they suffer from necrosis, right? And losing a limb is worse than dying for many of these farmers because it puts them out of work and they are the primary breadwinners of their families many times. So there's a lot of variation across venomous snakes in India, and we have looked at almost all the medically important snakes. This is a popular science kind of a talk. I'm not going into technical details. Happy to discuss with you them later. But just wanted to highlight the extent of variation and what levels you find these variations. If we go out on this campus, I'm pretty sure there are lots of cobras here, right? So if we collect venoms of the same species of cobra, let's say 10 cobras, do you expect to see any differences in the venoms, or do you think the venoms will be very similar to each other? You expect that they'll be similar, right? Because same environment, same kind of habitat. So we did this exercise in West Bengal where we collected venoms of this another species, non-big four species called a monoclonal cobra or monocellate cobra. You can see that unlike spectacle cobra, which has this V or U kind of shape, it has a circle, right? So we collected venoms of these guys in a very small radius, and we looked at the venom profiles. Now look at these donut charts. No two donut charts look similar to each other, right? So they varied so much that some snakes could produce venom that was 3 times as toxic as the other. And the antivenoms, Indian antivenoms didn't work very well against them because we don't manufacture antivenoms against monocellate cobras, which is responsible for a lot of deaths and disabilities in West Bengal, Orissa, a lot of eastern part of India, right? But we don't have an antivenom for that. But when we tested an antivenom that is made by Thai Society, Royal Society in Thailand, which they make against monocellate cobras there, it only worked against some individuals, but not against others. So the outcome of therapy can even depend on which individual snake that bit you. Can venom change within the same individual as they age? So we housed over 200 Russell swipers from the day of their birth, Russell swipers and cobras, and we looked at how their venom changes across time. We tracked that until they reached adulthood. Now look at the tail of the snake. I don't know if it's. Can you see it move? So it's wriggling. It's like a worm, right? It's doing that to attract amphibians and lizards towards it because they feed on these guys when they're very tiny, and they become older. It will be like a small snack, right? So they stop feeding on lizards and they start feeding on really large rodents. So this is called caudal luring, right? So when we looked at the venoms of these guys across their age, you can see how complex the venom becomes over time. Now, what do you think? Looking at this is very simpler, right? As soon as they're born, the venom is very simple, but when they get older, they produce so many different types of toxins. If you had to guess which of these two would be more toxic, the young ones or the adult ones? Younger ones? Yeah, logically you would think that probably it is this because it has so many different types of toxins, right? But when we tested this, I think I forgot to include that slide, but when we tested this on mice, we actually found that these guys are much, much more toxic, and that's because it produced very tiny amounts of them. And what is interesting is if you inject very limited amounts of venoms of the Russell swipers into mice, you will see the slide. I'll show you a video how quickly it kills mice. Um, but when these the venoms of these guys were injected into lizards, as I told you, they don't feed on lizards, right? So those lizards are happy even 24 hours later, nothing happens to them. But if you inject a tiny amount of the venom from the babies, it kills them almost instantaneously, right? So this shows how venom is very specific to what they feed on. So what is the consequence of all of this? There's a lot of variation even across seasons and so on, but I'm not showing any of that. But what is the consequence of all this variation? The consequence is that it has a negative impact on the effectiveness of antivenoms. In some places, it doesn't even protect. So the way we test this is we inject the venom into mice, and we also inject the antidote, and then see whether they survive or not. Statistically, significant numbers. And when we do that, some of these venoms or some of these antivenoms, they just don't protect mice injected with that venom at all. So it shows how ineffective they could be against some species. So I'll tell you what are the solutions that we are uh, producing for treating snake bites. And snakebite is not just India's problem. It affects a lot of developing countries around the world, including sub-Saharan Africa. So we thought, despite all the variation that you see, we thought, let's make a universal solution. Let's try to harness antibodies and develop antibodies that can knock out some of the most important toxins in these snakes around the world, and see if this antidote can be very effective across continents. So the toxin that we targeted was 3-phenyltoxin, which is the toxin that I told you earlier about. Makes up a majority of venoms of a lot of snakes, from black mambas to cobras in India and Africa, and crates, of course. So I'll not get into this, but the way we do this is there are different strategies that we follow. One is called yeast display selection. So basically, what you do is you harness the PBMCs from immunized animals. So we immunized, camels in Bikaner with toxins of our interest against which we wanted to make antibodies. We immunized cows and baboons, not in India because I didn't want to get lynched. We immunized them in Africa, and you can also get these cells from humans, right? Human snakebite survivors. You take those PBMCs and then you convert them into a library of yeast. So each individual yeast will display these antibodies on the surface, and then you can use antigens like viruses or venom components to pull out those antibodies that are very specific to the antigen of your interest, right? And then you can affinity maturate them and make them much better. So the first example that I'm going to show comes from a study like that, where we used naive human library. Actually, you take the PBMCs from humans who have never been bitten by snakes, and you convert them into a yeast library, and then you try to look for those antibodies that may opportunistically be very similar or be capable of binding to the venom component, right? The advantage of this is a synthetic library, meaning that it will have many more antibodies that you will not even find in nature, right? So I'll show you how useful this tool could be. I'm skipping a lot of slides, just showing you the ultimate results. So this is a snake from Southeast Asia, a crate, very dangerous snake. And one of the toxins that we got from this snake is responsible for deaths, in snake bite victims. So if you inject this toxin into mice, all of them die within 4 hours, right? But the moment you mix this toxin with this particular antibody that I showed you, you can see that all of them survive beyond 24 hours. Right? And we tested this against snakes from different parts of the world, including the monocellate cobras in eastern India, black mambas in Africa. Works perfectly against all of them, and it is also it also works in rescue experiments where you inject the venom first and then give the antidote. And why it was a big thing was because while we have antivenoms that don't even work across shorter distances, we now have solutions that can work across continents, and this is the first antibody that does work across such a large scale. Those efficacious because you now can produce them in mammalian cell lines, so you can mass produce lots of these antibodies, and instead of maintaining thousands and tens of thousands of horses for producing antivenoms every year, you could just produce them in bioreactors. And as I told you, these are human antibodies. If they are from cows or camels, you can humanize them. There are ways to do that. And then this is also ethical because you don't have to harm animals to produce them anymore. So this is actually a snake that, this paper was published on the cover, and this snake I photographed probably 5 minutes from my lab. This was from our campus on IIC, so I was happy that it was on the cover. I'll tell you a second example, a very quick example of another antibody that we discovered against Russell swiper, which is probably the most dangerous snake in the world, kills a lot of people. kills more people than any other snake species anywhere and causes a lot of disabilities too. So if you produce an antivenom or an antidote against this particular snake, you would probably solve the snake bite problem in its entirety, because this is responsible for almost 70-80% of cases in the country. And so we used the same technology that I showed you, but these libraries come from immunized camels. So convert into yeast libraries, and then we screen them against toxin of our interest. In this case, something called SVMP, snake venom metalloproteinase. And so this antibody is called 10x. So now I'll show you a video of what happens if you inject venom from this particular Russell swiper into mice. So look at this particular guy. I mean, this is 2 minutes after injection, right? Just 2 minutes into injection, it just collapses. And this, sometimes you actually start seeing mice dying. The minute you inject venoms, within a minute. So the venom is really, really toxic, right? And look at these mice which were injected with the venom and antivenom. Sorry, the particular antibody that we discovered. And this is 24 hours after injection, right? They're doing perfectly well. So this shows you, despite all the complexity in venoms that I talked about, if you knock out one particular toxin, both examples, right? In the previous example, we knocked out the three-finger toxin. Here we produce the first, SVMP, anti-SVMP antibody in the world, and this is also targeting just one toxin, and you achieve complete protection. So this shows that despite the complexity in venoms, you could, harness these recombinant antibody technologies to probably produce much better antidotes for snake bites. And we have tested this against snakes across India, Russell swiper populations across India. I'm showing some of them here. It works everywhere except in West Bengal, and that's because snakes there produce a different type of toxin along with SVMP called PLA2, phospholipase A2, and we have antibodies against that toxin as well. So I think that's my time. Thank you so much for your attention. That's my.
I must compliment you for excellent presentation.
Oh, thank you.
Yesterday we were talking about the translation value of research, and this is what you have showed, that how much valuable research is being pursued. My question is that we have a person right in the name of Professor K.K. Sharma, probably you might be knowing, and he claims that we should have a snake bite death-free India. And also raises question about CPCSC intervention, about use of camel or use of horses in immunization and developing antibodies. So did you come across any such thing? And if yes, do you have any suggestion where such a big thing, where thousands and thousands of people dying of the snake bite, so we don't want to kill the snakes in the interest of the ecosystem? At the same time, we want that can we develop anti-venom preparations which could be used for people who suffer snake bites.
Yeah, answering the first part of your question, I think it's impossible to have snake bite free any place in the world, especially India, because there's so much of human-animal encounter, right? I mean, because of that, human densities as well as snakes, which like to live around human settlements. And they are very cryptic. If I take you out for a walk, the snake might be there and you probably will not notice it because it's very well camouflaged, right? So you only need people who are experienced in looking at them, and people who work on the ground, they don't have time to look for snakes. They already have so many issues in their life to deal with. So that's why I don't think you can have snake bite free place. But yes, definitely, these antibodies are definitely one solution. But I didn't include a lot of other things that we are developing, including small molecule inhibitors, which you can take like crocin pills in the field as soon as you're bitten by a snake, and we have shown that it works beautifully against Russell swiper. But again, there are like different combinations of these molecules that you can take, and we have made like a map. So if you're in West Bengal, you take, let's say, red pill versus blue pill, and if you're in Karnataka, you take a completely different pill. But all of those are orally administrable. So I think small molecules would be the future if we have developed cocktails that work very effectively in different places. But collecting venoms, as you said, regulatory issues are there. Some of the permissions that I had applied before starting my lab in 2017, I've gotten them now. Some of them I haven't even heard back, despite talking to them almost every now and then. So yeah, I mean, if regulatory issues are sorted, then But yeah, coming to the animal ethics problem, as I said, if you can produce recombinant antibodies in bioreactors, you don't have to immunize animals anymore. So we immunize 16 camels, and whatever antibodies we have got now, if they clear clinical trials, you could mass produce them, and then you don't have to immunize any animal anymore, right? Or at least for producing antivenoms in India.
Yes, please. I have a question regarding the vaccination. Whether it's possible vaccination in future?
It's very difficult because vaccines, so for example, if you're bitten by, sorry, not bitten by, I'm used to saying the word bitten, but let's say if you're infected with COVID, you have enough time to mount an immune response, right? Your body has enough time to mount that. But if you're bitten by a snake, as I showed you, the toxins they can cause irreversible damage in a very short time. So your body won't have.
You can give an advanced in suspected population.
Prophylactics is one option, but it doesn't sustain very well for a very long time, and I think that would also be very expensive. So, but definitely that's an area we are also looking into.
Yes,
Wonderful talk. I just want to make a point that can it be possible to have biosensors or sensing because prevention is better than cure, and then if
Oh.
we can detect them.
Detect snakes?
Yes.
Okay, I think that's very difficult because I mean, we can't do that for tigers, right? I mean, there are very fewer tigers in India than snakes, but yeah, doing that for snakes is very difficult, I would assume. But you do need a detection kit. Biosensors, we are developing those as well. So in fact, this antibody is very useful in detecting as well, because when a snake bites somebody and they go to hospital, the clinician doesn't know what snake has bitten them. Even if you bring a snake, they will not know. So we are developing, detection kits like pregnancy kits, that work in 2 minutes. So we have an excellent prototype for even detecting snake bites, at least for us as well.
Wearables can also be a better bet for protecting.
I am not sure if, I mean, possibly, I don't know. I'm not an engineer, so I can't comment, but I think it would be too.
Thank you for your excellent talk. Very interesting topic. It's for the young researchers. How did you get interested in this?
Oh, I saw a snake and I picked it up. No, don't do that. I was very interested in snakes. I read about them and I started handling them. I was a rescuer, and that's how I got it.
Thank you.
I think there are some kind of traditional methods
Yeah.
of preventing snake community.
Those don't
They
work.
put some water.
Yeah, no, those don't work. I mean, they also say you plant this tree or that tree or put kerosene. Kerosene basically just burns them. So it's not very nice for those animals. But
No.
yeah, they don't.
Snake venom is used for drugs.
What?
Drugs.
Oh yeah.
Anti-platelet drugs.
Yeah,
So
absolutely.
how is that market going on? Where is
Yes.
that?
So we were talking about strokes and other things, right? So if you know anyone who takes medication for high blood pressure, it's actually a modified snake venom toxin. It's a multi-billion-dollar industry now. Captopril was a drug that was introduced first, and there are lots of those drugs being developed for diabetes, from Gila monster, lizard venoms, and so yeah, and we have also found a lot of amazing compounds with therapeutic potential, like the wasps that you see flying around. We have found something that prevents blood from clotting even 40 minutes after mixing with the blood, and that's the upper limit of the detection. So I think there are lots of amazing drugs that are waiting to be discovered from these animals that we see all around us, and we.
Venom beer. Snake venom beer.
Beer. Okay, I'm a teetotaler. I'm not probably the best person to comment on it.
Just Google. You just Google, the beer, snake venom.
Maybe without the mic, I am Dr. Rawat. Basically, I am a microbiologist. So I was just interested, venom is a modified saliva, and saliva is the quality of saliva depends upon the microbiome. So any studies is there a difference in the bacteria which are colonizing the salivary glands of these snakes, and whether they are having any effect on the heightened venom?
You do find bacteria in mouth of snakes, but those are mostly they come from feces, because when they eat mice, they eat them whole, right? So they defecate and urinate. So it mostly comes from there. But these toxins that they produce, they are there in our saliva as well. We are using them for immunity and reproduction, but in our immune system and all, the same components are there. They are not using anything different. So it's just remarkable how they have modified these completely non-toxic compounds into some of the most potent proteins that we know.
Yeah, for snake venom studies, any regularities?
Oh yeah. We need to get permissions from state forest departments. Then, so I'll give you an example. I need to first contact for Karnataka Forest Department to get, let's say in Karnataka, get an approval from the boss, and then I need to go to every single district and sign a MOU with each one of them, which sometimes takes a long time. And then nowadays I made them scheduled to a species, which means that an approval also needs to be gotten from the central ministry. So it takes a long time, which is also why I think there are not many venom labs.
Okay. We'll wind up the session. Last
My question is very similar to the Rahul about the technical preventive measurements.
Yeah.
I think villages are using people's mix ammonium chloride in mustard seeds, by pollen no other. They distribute their area and then they think that it's a preventive measure.
Unfortunately, See, mice. Why do snakes come close to people? Because they feed on. Many of the snakes that bite people are the ones that feed on mice. Where do you find mice? Around humans, right? So, if a house is not clean, if you're getting snake bites within cities and all, if a house is not clean and you're getting a lot of rodents and stuff, that's where you will find snakes. So, unfortunately, there is no countermeasure to prevent them from entering your houses. But if you leave them be, and if you don't step on them, if you don't go closer, I think that's probably the best preventive measure you can take.
Okay, we will end the session. I really thank all the speakers for a wonderful session, and the first speaker, Professor Cruz, he gave the